Hi, everyone. Thank you very much for joining me today in this incredible experience of XPANSE 2024. I'm very happy to share with you our advancement in cell labeling, a technology that can open up new opportunities in biomedicine. Imagine a healthcare platform thanks to which you can follow and track, visualize cells in our body with an exceptional precision. Let me start explaining this concept. Think about packages being shipped. And thanks to a barcode, a unique identifier, we can know what is inside and their destination. Well, in the same way, we can use a barcode, an innovative barcode, to tag cells and to follow them, to know what is their role, to know their function in our body. So I want to say that these barcodes should have specific characteristics. They should be ultra small, they should be biocompatible, and of course, we should be able to identify them thanks to specific technologies. In this context, the green fluorescent protein, which was discovered on a jellyfish, was an incredible step to visualize cells, thanks to fluorescent-based technologies. This was good, was an incredible discovery, worthy of a Nobel Prize. However, there are limitations. So what are those limitations? Well, for green fluorescent protein, it is difficult to think their use in humans in vivo because of toxicological and ethical issues. And also, on top of green fluorescent protein, we have a very limited number of fluorescent molecules. Therefore, limiting the multiplexing and so the information we can gather from single cells in a complex cellular environment, which is the one we have in our body. So to overcome this limit, we used a technology that is not based on fluorescence anymore. It's based on the use of metals and is called single-cell mass cytometry. So here, metals are tagged with antibodies. Each antibody can link to specific cells, forming an ion cloud where heavy metals can be distinguished by a time of flight, a ratio mass charge. So each color will identify a peak corresponding to a different cell surface marker. So this is a powerful technology. However, the tags we have in single-cell mass cytometry, none of them is biocompatible, and still they lack of versatility. They lack versatility. We don't have enough chemistries for the biological, medical questions we have. And so this is when I realized that actually MaxSense, a material discovered by Yuri Gogotsi, had all the potentials to join the match against traditional cell labeling tools. MaxSense are transition metals, carbides, and nitrides, And what makes them unique is that they have a very versatile chemistry. We can play with them, we can design them in a composition, in a chemical composition that can allow us to make them detectable on single-cell mass cytometry, having an incredible opportunity to color and label cells. So this was a work we published where the data were truly encourage us to transform the current vision on vaccines into max tags, to color, to label cells with an incredible precision. Here is an overview of what we have done. As a proof of concept, we used the periodic table of elements, choosing specific transition metals, molybdenum, niobium, tantalum. We performed several experiments on human cells and mice, and then we were able to track their presence thanks to 3 different single-cell mass cytometry platforms. Here you have the site of range at the top, where each peak, each color, corresponds to a cell marker. As you can see, those are commercial tags. As you can see, the commercial tags leave several space empty, white space. Indeed, just with those 3 vaccines, we were able to use unexplored channel. But not only that, we know that we can play with their chemistry to fulfill all the other empty channels and therefore gather more information on the cells. But not only that, we can actually go well beyond this site of range, playing with the ratio of metals in the single vaccine. On top of this, in this study, we followed simultaneously the expression of over 30 cell markers in humans and also in mice. So, just an example, for instance, we saw the presence of 3 different vaccines in 15 immune cell types, which is a truly in-depth immune profiling. We were also able to track them up to 14 days. This is, I will say, amazing if we think about the possibility of long-term cell tracking. And well, we now have an in vivo prototype. We can take cells from mice, we can treat them with vaccines. We tried that with several types of vaccines. We can reinject the cells in mice, and we can follow their fate, their destiny, in multiple cell types, tissues, and organs. So what if we can give this tool to clinicians that are struggling in knowing where cell goes after cell-based therapy that include a transplantation. So we transformed vaccines in max tags, powerful cell labeling tools. They have an incredible versatile chemistry. They have no noise, no spectral overlap, of course, no autofluorescence problems. The cell labeling market was valued at $8.6 billion with a compound annual rate growth of 16.5. This number truly can tell the high demand there is in the clinic for new advancement in cell labeling, in tracking, in visualize cells. And what we have done is to designed to manufacture something that is going well beyond a cell labeling tool. It can visualize, it can light up life itself. So, having said that, I truly hope this can be of benefit for an innovative future in healthcare. I really thank you, all of you, for your kind attention, incredible audience of XPANSE, and thank you, Dr. Zina Chinkar and EDQ Abu Dhabi. Thank you.